Okay, let's go. Let's get going. So, when I was looking at the syllabus, what I came across is some of the stuff we haven't covered yet. That is how to measure the mutation frequency or rate of mutation. Then we have mutator strain, and, concept of mutator strain. Before going into the measurement of mutator, concept of measuring mutation, either the mutation rate or frequency, let's go ahead. What we different types of mutations that we can think about. One is kind of uh, point mutations that we can think about, like when there is a mutation where it's a change in a single nucleotide, it can change the translational frames, which can give rise to kind of, even if there is a nucleotide change, it can still code for the same amino acid. That's called the silent mutations or same sense mutations. So generally it's called silent mutations. Then in case of missense mutations, what we see is that the change in the nucleotide, either is a single or multiple, changes the uh, amino acid that is caught by those uh, uh, codons. So there is a change in the amino acid in the coding region due to the change of nucleotide. That's called the missense mutations. When the change in the nucleotide leads to the formation of a translational stop codon from a coding or sense codon to the non-coding or stop codon, that is called the nonsense mutations. So depending on the translation results of the mutations, we can have three mutations. One is there is no change in the amino acid, that is called silent mutations or same sense mutations. Then you have missense mutations, which changes the amino acid, for example, from glycine to proline or histidine. So there is a change in the identity of the amino acid that is coded for, or it leads to something that doesn't code for anything, or doesn't code for anything means stop codons. Okay, those are called nonsense mutations. Depending on the type of changes in the nucleotide identity, for example, the change in the nucleotide can be pyrimidine to pyrimidine, or pyrimidine to purines, or purines to purines. So depending on this, there are two types of mutations. Transitions. Transitions is the purines to purines or purine to purine mutation change is called transitions. And you have transversions. Those are pyrimidines to purine or purines to pyrimidine kind of change in the nucleotide. Then we have, depending on the, when we discussed earlier in the DNA repair classes, what we discussed is that there are repair mechanisms which are very, very much error prone, and that can give rise to the addition of nucleotides or the removing of nucleotides at the repaired positions. So that would change how the amino acid is being, or when an mRNA is being decoded to a protein translation system. So depending on that, this is just repetition of what we were talking about: silent, nonsense, and missense. So the First one is the, for example, at the DNA level, TTC codes for, and mRNA level, AAG codes for lysine, suppose. Then, in the silent mutation, what happens is the identity of amino acid doesn't change, but the codon is, or for example, G is changed to A due to a point mutation. This is called a silent mutation. And if there is a change in the single nucleotide or multiple nucleotide, which leads to the formation of a stop codon, that is nonsense mutation. And in the case of missense mutations, when there is change in the amino acid identity, it can be something like conserved or non-conserved mutations. What do you mean by conserved or non-conserved is that there is not much change in the identity of the amino acid, and it can also change to something like which is not related. For example, when you have change in the amino acid with similar site groups, those will be conservative, and if there is a totally change, then it won't be non-conservative mutations. So going ahead, I talked to you about transitions and transpositions, purine to purines or pyrimidine purines. These are transitions, and if it is changed from purines to pyrimidines or from pyrimidines to purines, those are called the transversions of mutations. Now, when I was telling about frame shift mutation, this is one good example here. For example, in the starting, the codon reading is in the frame, what I box here. AGC is one codon, GTZ is second codon, and AAA is third codon, AAT is fourth codon in the starting point. Due to some changes, for example, if insertion or deletion of one of the nucleotide, if you look at it, now changes reading frame from the, in the case of starting, it was AAT, now it is changing to GTC triple ATT. So basically it changes the amino acid, okay, because of the mutation. So in the case of this is a one base deletion. So when you have that basically change the reading frame, and again insertion also is going to change the reading frame during the translation process. So frame shift mutations, missense mutations, silent mutations, or nonsense mutations, all these are definition from the side of translational during the translational result. Okay. Then when we talked about these changes in mutate, or these changes can be either substitutions, deletions of one or longer base nucleotides, can be through the it can be measured in certain ways. So if you take any organism, there is a certainty that there will be mutation, but how often or how frequently the mutation is generated is very, very interesting. For example, when we talk about viruses, for example, HIV, so far we cannot get these infections because the genome of HIV keeps on mutating very fast, and whatever vaccine which was supposed to work is changed in the next generation of virus infections. So basically one type of vaccine is not going to work in for a longer run. So the frequency of mutation has very much relevance in understanding the evolutionary part, or the in the treatment or understanding of a disease causing organism, or resistance to certain types of antibiotics. All these mutation frequencies are very much important to uh, know about it. So there are two methods to measure mutation rate. So mutation rate, when we say, and we'll come to that point. So mutation rate, the two methods generally utilizes is the mutation accumulation. So what you do is you allow the organism to grow for a certain period of time and look at the overall mutation which is being accumulated, and you see, and you average out and then the calculate the mutation frequency from there. So basically you have a single culture, and in this mutation accumulation calculation, and in the case of fluctuation analysis, you are going to check whether the frequency which is the mutation which is happening randomly is how often, what is the probability of getting this mutation. So to go ahead, the fluctuation test or fluctuation analysis that we are talking about here comes from the initial experiment from Luria and Delbruck, and in taking their name, the test is called the Luria-Delbruck fluctuation test. What do you do by this fluctuation test? First, what basically it does is that you have a culture of bacteria in this case, or in their experiment they have checked the mutation which leads to the resistance of a bacteria to bacteriophage. So in this case, what you do is a culture of bacteria is treated with bacteriophage and allow it to evolve in such a way that it is resistant to the bacteriophage. That was the experiment they did. But the experiment, what is being done here is that initial inoculum or parent culture, where we expect that there is no mutation, is subculture in a very diluted condition, in such a way that you assume that there is one bacteria per unit volume. For example, if you are doing some culture of 10 ml, you expect that per unit you can say one bacteria per 100 microliter, or one bacteria per 1 ml. So per unit volume you have a single bacteria, and you inoculate it and culture and plate in such a way to check whether any mutant which is going to be resistant to bacteriophage or resistant to antibiotics, you plate on a selective medium plate. So you have these experiments run in parallel. You have to have in parallel just to make sure that the mutation is happening random. How do we know that? So in this case, in multiple parallel cultures, if it is happening randomly, what do you expect? It is that the mutation is going to be, the number of mutants or resistant colonies is going to be different. But if it is not random, it is due to intrinsic property of or condition of the culture, the number of colonies what you get is going to be similar. Okay, so the Luria-Delbruck fluctuation test is about running parallel cultures and look at the number of colonies you are getting, and then using this number of colonies and number of parallel cultures you can calculate the mutation rate. How to calculate, we'll discuss. There are many methods that has been discussed. In the accumulation test, what we were talking about, you can you can grow a single culture and look at the calculate the per volume number of resistant colonies or mutants. So if we look this experiment, what we can say is that if something which is not random or spontaneous mutation, what you expect is that number of resistant colonies are going to be same in all the parallel sets of experiments or parallel cultures you are maintaining. Okay, but if it is spontaneous mutation, it's a mutation is happening randomly. What we expect is the random mutation can happen at the first cell division, or at a second cell division, or third cell division. So there is a huge difference in here, because if the mutation happens in the first cell division, the first one of the daughter cells is going to be mutant, just as it is shown. Well, if this is a mutant, then all its offsprings are going to be resistant to colonies. So in this case, we have 4 resistant colonies after 3 cell divisions. 1, 2, 3, yes, after 3 generations. But if it is happening at the last or third cell division, then only one of them is going to be resistant. You will get only one resistant colony. But if it is happening after the second cell division, only one colony is only 2 colonies are going to be resistant. In fact, what it does tells you that for each parallel culture, you expect different number of mutant colonies which are resistant to bacteria or antibiotics that you are testing for. So in certain scenario also, there may not be any mutation in all the 3 generations or in the 3 cell divisions that you look for. Those will not give any resistant colonies. So basically, it will give you a different number of resistant colonies in case when it is a spontaneous mutation. But if it is because of certain induced or because of certain reagents or conditions which is present in the environment which induce mutations, then you are likely to get similar number of resistance colonies in here, and it will be very random. Okay, so then next is, so this is what we were discussing about. If the mutation, if you start at the generation 0 is a mutant, then you will have 8 colonies of mutations. Then if it is in the first, it will have 4. If it is in the second generation, it will have 2. If it is in third generation, you will have 1. And if in the fourth generation, it will be 0. Okay, so that's how you're going to get different number of mutations depending on when the mutation happened. So here, if you look at it, total number of mutants is 1, 4, 12, and 32, depending on at what time the mutation has happened. And the fraction of, if you look at it, if you start with, if you have to calculate the mutation frequency, the total number of mutants, and then for each generation, what kind of mutation, how many mutations, and for each generation, how many mutations have occurred. That will tell you the rate of mutation or frequency of mutation for each generation, correct? So in this case, mutant fraction is R by N, total number of mutants divided by total number of cells in that generation. So in the case of here, 0, 8 by 8 out of the total number. So you can see the change in the mutation frequency. So now we need to, before we go into the technicalities of calculating mutation frequencies, we have few terms. One is M is the number of mutations per culture. Then mu is the mutation rate. So both of these are very, very relevant when we look into mutation rate or mutation frequency in a population. For example, in a bacterial population or even in human population, both are equally applicable. Correct. So M is the number of mutations per culture. Mu is the mutation rate. Here N is the number of cells. N0 is the initial number of cells at the start of the inoculum. And NT is the total number of cells at the end of the experiment. And you have observed number of mutants, then median number of mutants, that is R. F is the frequency. Okay, so these things are very important. P0 is the proportion of cultures without mutants. Why these definitions are important? Because few of the next few slides, I'm going to discuss how to calculate number of mutations. And after calculating the number of mutations, you will look for the mutation rate. So one of the first mutation frequency calculation, there are different models depending on the conditions that people have used. And one of the popular one of the popular model to calculate is the Lee-Corson model. Okay, so in this, which is also equally applied in the Luria-Delbruck fluctuation test, which is very much used now also, is that the cells are growing exponentially. Okay, if it is a stationary one, these models are not going to hold. Fluctuation analysis is not going to hold. Okay, and then second thing is the mutations are independent of each other. So one mutation doesn't influence the next mutation that is going to be obtained. Then third is that the mutation is constant throughout the cell lifetime. Okay, there are many assumptions. So all these things we don't. If you look at it, and depending on the timing of the mutations, also the model changes. But these assumptions are equally applied when you are using fluctuation test. So one of the method to calculate the mutation number is called the P0 method. This is one of the most popular P0 method to calculate mutation number. Okay, mutation number is how you have to calculate is P naught is equal to e to the power minus m. So P naught is the proportion of cultures with no mutations. So we were talking about this kind of experiments here. So for example, in this one, we are using 5 parallel cultures. P naught will be 1 out of 5, that is 0.2. Okay, P naught will be 0.2 for this figure. So likewise, you can know P naught. P naught is equal to this much. For the experiment that I just saw is 0.2, P naught is 0.2 is equal to e to minus m. So m can be calculated very easily. M is equal to minus natural log of P naught. So you can find out m for this. But this P0 method of calculating is very easy, but there are few limitations that we can discuss in the later slides. And one more thing is that if you are not plating every culture or all the cultures, for example, if you have a culture of 10 ml, but if you are plating only 100 microliters, then the M value changes. So how to adjust for that partial plating when you do experiments? Many times this kind of plating the whole culture may not be uh, viable. In that case, how to correct it is M actual mutation number of mutations actual is equal to mutation which is observed. That is what we calculated here. This is the observed M value. G is the fraction of plating and GLNG. With these corrections, you can get very good estimate of actual mutation number of that culture. Okay, so this is one of the most popular, one of the popular method. There are many other methods. For example, another method which is being used in by other people is that accumulation of clones method. This is there. So in that case, the calculation of n is pretty much different from the P0 method. But I would not like you to focus in this method for the time being. There are so many methods. I'm going to attach a paper wherein they discuss different types of methods. Some of them are quite complicated, so you can have a look, but not for exam very complicated question. But the P0 method definitely is very popular, and it's important to know that method. Next, again for this accumulation method, the calculation for the mutation frequency you can use this kind of method. In case when there is a very simplified way of calculating, what you can do is that, because at any point of time, what you can find out is that the mutation frequency multiplied by the total number of cells would give you the mutation number, correct? So from there you can calculate the basically the mutation frequency is equal to mutation number divided by number of cells which was there just one before the actual division. So when the n total is very big, you can assume that n total minus 1 is more or less the same. So in that case, mutation number divided by total number of the cells at the end of the culture would give you mutation frequency. This is very, very simplified calculation. So this kind of mutations, if you think this mutation frequency calculation, okay, mutation frequency calculation is applicable when you assume that the mutation happens during replication or during cell division. Okay, so this simplified form mutation frequency is equal to mutation number divided by total number of cells is applicable when you assume that mutation happens because of or during cell division. But if the mutation happens before the cell division, before the replication happens, then because of some mutagens or something, then you need to modify the calculation of mutation frequency to mutation frequency is equal to m by 2 into nt. So this is the one which is need to be used. Okay, so depending on the timing, you need to modify timing of mutation. If it is before replication, before cell division, the numerator becomes much bigger. Okay, so and then next, most of our cultures that we use, E. coli or all these things, doesn't grow synchronously. What does it mean by synchronous culture is that all the cells are at the same stage of cell cycle. So if replication is starting, that means all the cells in the culture have started uh, replication. When it is dividing the cells into two daughter cells, all the cells are dividing into two cells. That is called synchronous culture. But most of our cultures in the experimental setting that you will do for antibiotic resistance or anything, that are asynchronous. That means all the cells are not at the same stage of the cell cycle. Some are at the initial DNA replication state, some are dividing, some are just finished dividing. So all the cells are, many cells are in different stages of the cell cycle. In that case, the mutation frequency again changes further. So mutation frequency is equal to mu mutation number natural log of 2 divided by total number of cells in that culture. So it's equal to natural log of 2 is 0.6932 into mutation number divided by NT. So depending on the growth characteristic, whether it is a synchronous culture or asynchronous culture, you have to put in these factors in mind. So why it is important is that depending on the formula you are using, the uh, the mutation frequency number, okay, is going to be different. And they are in the ratio 1 is to 0.5 is to 0.693 in the three ways of calculating mutation frequency that I just discussed now. Okay, so there are three different ways for a simplified calculation of mutation frequency itself. So now what I have been talking about is there are different types of calculating mutation numbers depending on the model. The P0 method that I focused earlier applies very well or holds true when M, the mutation number, is in between 0.3 and 2.3. If it is below 0.3, again, it's not good. If it is above 2.3, again, it is much less reliable. So what we talked about is the mutation number, M, is equal to minus natural log of P0. That is the P0 method of calculating mutation number, which can be utilized, that M can be utilized for calculating mutation frequency. Then there is another very popular method that is called the method of the mean. In this case, it is very flexible, it is applicable to many, and in this case, what we assume is that, what we were talking about is that mutation frequency into total number of cells will add that much number of mutants in the next generation. Okay, so so mutation number in one generation you can calculate. M is equal to so basically you have to calculate the number of mutants in one. Suppose it's in the third generation, you will find out the number of mutants in the third generation. Then you go to the fourth generation, how many mutants are there in the culture. So if you look at it, the difference between, in the case of third generation, if you get 5, suppose, then in the fourth generation, you have 6 of them. So that means the difference in the number is 3, correct? So that 3 is equal to mutation frequency into the total number of cells in the third generation. So that will give you mutation frequency or mutation number as well as mutation frequency in that in that culture. So another one that I just started out, Lee-Colson method of the median, that is another way, and this holds true only when M is, or mutation number is between 1.5 to 15. Okay, so that can be calculated using this formula. Number of mutations that you observe then is equal to, using this formula you can calculate the m value. And another very popular one is the drag formula. Drag formula is applicable when m is greater than or equal to 30. So in this case, the mutation frequency is equal to the frequency you are observing divided by natural log of total number of cells into frequency. So if you calculate this formula, you can get the m values out of it. So, so far what we have been talking, all the methods for calculating m or mu here is that the is the fluctuation analysis. But in the case of accumulation analysis, what we can use is taking into consideration that the culture you are dealing with is a continuously dividing culture. Then mu can be calculated by the number of mutations you has observed in the second generation minus or nth generation, then the mutation frequency or number of frequency divided by this formula. So you can use this for using the considering the accumulation way of calculation mutation rate. Okay, so all these experiments, the frequency of mutation in the nth generation can be found out, F1 can be found out. All this can be found out using the experimental parameters you have. Okay, so then what we were talking about is that in the case of accumulation frequency, what you're basically doing is that you allow the mutation to accumulate after enough number of generations. For example, in this case, each individual in a population, and you can allow it to accumulate, and after some time you calculate all these things compared with the ancestral sequence and then see how much mutation has been accumulated, and from there you can calculate a mutation frequency. Okay, so that is the two methods of mutation frequency calculation. Next, what we need to do is the mutator strains. So what do you mean by mutator strains is that the background, the strain which is highly mutagenic or due to mutation in certain genes give rise to enhanced rate of mutation. That those strains carrying those mutations are called mutator strains. Now, for example, the mut-D homolog, DNA-Q homolog of the part of the DNA polymerase tree. DNA polymerase tree is the one which is involved in DNA replication, correct? So DNA replication complex, if there is a mutation in mut-D, it enhances the mutation rate to 1000x. So the strain which carries mutation in mut-D will be a mutator strain. Then again, in the mismatch repair, in the repair system mechanisms, if there is a mutation like something like mut LSH, any of these genes, if it is mutated, it can enhance the mutation rate to 100x, and these are one type of another mutator strains. So there can be mutator strains can have mutations in the replication machinery or DNA repair machinery, or there can be other metabolic pathways which affects overall the efficiency of mutation or the fidelity of repair or replication can also become a mutator strain. So here is one example. This is basically picked up from a research paper itself. What you are seeing here is the organism wild type is looked into for its mutability, under the treatment of uh, tetracycline, rifampicin, or streptomycin. These all affect slightly different pathways. So in the case of tetracycline, if you look at it, wild type, deletion of mut T or deletion of mut Y, there is hardly any change in these three strains. But if you look at rifampicin, what you see is that mut Y mutant is much more mutagenic as compared to wild type. In the case of streptomycin, mut is much more mutagenic as compared to the other wild type or the mut-Y strain. So basically, depending on the stress, it can give rise to different types of mutation, and all these strains can be considered as mutated strains under certain conditions. This has been seen in the case of eukaryotes as well. This example comes from the study with cerevisiae. So what the people have done is they have looked into the uh, mutants which are involved in, suppose, DNA polymerase, Pol3 mutation, MUSH. Again, this is homologous recombination. If you attended Nissan's classes, MSH is very much there in his lectures. And then RAD, again, another pathway of uh, DNA repair. If you look at it, what it is seen is that the most of the mutations in wild type is single nucleotide morphism or changes in single nucleotide. Okay, then if you look at a mutant, something like MUSH, it is lot of small uh, insertions or deletions of nucleotides are seen again. And then in the case of together with MEG1 or TEL1 delta, what you see is that lot of aneuploidy. Aneuploidy means the number of chromosomes are changed, or there is a missegregation of chromosomes is happening very often in this mutant. So depending on the type of mutants you can have, they have different phenotypes, and all of these are mutative phenotype. Okay, so the occurrence, if you look at it, and here in B, it's much as compared to wild type, it's much different, much enhanced in many of these mutant phenotype. So these are mutator strains. Okay, so why these mutator strains are important is that if you want to do random mutagenesis, for example, if you take the mutated strain of E. coli, you take it, and many times what people do is you take the mutated strain of E. coli, throw in a plasmid which calls for some protein of your interest. You put inside the plasmid, let it replicate inside a mutated strain, because mutated strain would give rise to multiple random mutations. What you expect is that it will also introduce mutations, multiple mutations in the plasmid you have put in. So once you isolate the plasmid, what you will get is that it's the plasmid you are obtaining from the mutated strain is a collection of mutant plasmids. Indirectly, it's a collection of mutants of your protein of interest. So you can utilize mutated strain to generate mutants of your protein of interest. Now, if you have a conditional or a strain wherein you can test the functionality of your protein of interest, what you can do is you put this library of uh, plasmids into that strain and check which of the mutants are functioning better, or which of the strains are not, or which of the mutants are not functional well. You can differentiate the function of those mutants. Once you know the function of the mutants, you can isolate and check what is the mutation in that construct, and you can find out the functionality of that particular mutation. So you can do functional genomics using mutator strain itself by generating random mutations. Okay, so you can ask many different questions by using different types of mutator strains. Okay, so if you want to look at the mechanism of aneuploidy, then maybe you may be able to take MEG1 and TAL1 deletion mutant and see how does the chromosomes are affecting by each of the MEG1 strain or TAL1 deletion strain. Okay, so with this, we'll stop for today. And if you have any question, let me know. Okay, any question? Any question, guys? If there is no question, then maybe I'll stop here for today and see you in the next week. I think when I look at the syllabus, few more topics are there. So next week will be last, I think Monday and Tuesday. Okay, so I was in Wednesday and Thursday, so there is no problem. So next week, Wednesday and Thursday, and then we wind up the classes. Okay, thank you guys. See you.
Thank you, sir.